Hi everyone. Alright, so in this video, we're continuing talking about apoptosis. In our prior videos, we talked about the TNF fast signaling pathway, intrinsic apoptotic signaling. We had 2 videos on that. And now we are going to continue talking about apoptosis, but this time in the context of p53. Which is one of our major tumor suppressors in the cell that is going to be helping the cell go through the process of apoptosis. And p53 has a mix of activities when it comes to apoptosis. It's going to have kind of an array of transcriptional functions, which we'll talk about first. And then it also will have some cytoplasmic functions. So let's talk about transcription first. So when I say transcription, I mean that this is p53 acting as a transcription factor in the nucleus, impacting the production of various genes. And to step through the genes that it will have an impact on, the first is that it's going to increase the production of our pro-apoptotic proteins, BACs and BAC. So we want to have more of those pro-apoptotic proteins present to be inserted into the mitochondrial membrane. It's going to produce less of our pro-life proteins. So BCL2 and BCLXL, we're going to produce less of. This can kind of be lumped together under altering transcription of apoptotic factors. A third action is going to be that it increases the transcription of the FAST receptor. Now, the FAST receptor is important for initiating extrinsic signaling for apoptosis. So if we have more of our FAST receptor present, then we're more able to actually receive those extrinsic signals, because there's just a higher density of receptors on the surface of the cell, so more of the ligands will be able to bind. We're going to increase the transcription of something called IGF-BP, IGF-BP3, and this protein is actually involved in the p53 pathway. So IGF-BP3 inhibits a protein called IGF-1, which normally promotes activation of the PI3K pathway. So this is going to inhibit activation of the PI3K pathway, so we're down-regulating that pathway. PI3K is important for survival, proliferation, etc. All good things for cancer cells. So we want to prevent that from happening by inhibiting this pathway. The fifth is going to be increasing transcription of another transcription factor. So there's a factor called FOXO3, which is a transcription factor that we're going to produce, and this leads to an increase in other pro-apoptotic proteins, right? So that's kind of our fifth thing that we transcribe. And lastly, we increase transcription of something called PUMA. And we'll talk about that a little bit more in our cytoplasmic functions of p53. But these are our impacts on transcription in particular, as we're altering the levels of transcription for these various proteins. In terms of p53's cytoplasmic functions, let's address that right here. Cytoplasmic functions. I'm going to draw our mitochondrial outer membrane here. I'm going to put kind of a pro-apoptotic protein there. Maybe that's back. I'm going to put an anti-apoptotic protein here. And p53 can have a couple different impacts. The first is going to be that p53, which I'm going to label in green here, p53 is going to bind and inhibit BCL2 and BCL-XL. So it's binding and inhibiting our anti-apoptotic proteins, and this is going to allow for increased pore formation, right? So acting very similar to a death helper in this setting. Another activity that it's able to engage in, if we draw up here, just laying out what it's going to look like, is it can do the secondary death helper activity where it binds to back, inserts it into the membrane, and then p53 leaves. So this is the activity that we call kiss and run. So p53 can do that as well for our back and Bax proteins, inserting them into the membrane. And then we talked also about how PUMA is able to, or is transcribed by p53. The function of PUMA is that it is also able to bind and inhibit BCL2 and BCL-XL. So this is my PUMA protein. This is inhibiting this, just like p53 is inhibiting this protein. So PUMA can bind and inhibit BCL2 and BCL-XL, which I didn't label this, but maybe this is BCL2, and maybe this is BCL-XL. And that frees up p53 to be able to engage in more of this kiss and run activity, right? So that's kind of our p53 related activities for apoptosis. I'll make one other short video discussing some of the therapeutics for apoptosis and how we can kind of help this process along.